So I didn't know that I wanted to become a researcher right away. I knew that I wanted to do science. So my undergraduate degree is in biochemistry and molecular biology. It wasn't until I had my first job as a researcher that I realized that I really loved this. And all I was doing was just a bunch of PCRs in a lab, like repetitive work over and over. But I really liked the lab environment, and I liked that you had a problem that nobody else is working on at that moment. So every day you are learning about something new that nobody else has thought about before. When I'm driving to work in the morning, I have about a 20-minute drive. That's the time I start to weed through all of the technical problems that we're having. So all the students, we communicate on Slack. So whenever they ask me a question, I'll typically answer right after I get home from work because of that drive and on my way to work. So I really love working through those technical challenges with students and trying to resolve them in creative ways. So our lab is really interested in understanding RNA modifications. So fortunately, right now, a lot of the world knows what an RNA modification is because that's what the SARS-CoV-2 vaccine from Moderna and Pfizer is using. There's a modified RNA that's inside of that sequence that we're all injecting into our bodies, and it's helping that vaccine become more effective. But that modification is also occurring inside of the human body, and we don't really know why it's there. So our lab is developing tools so that we can figure out what it's doing inside of our cells, where it is inside of the cells, and what the functions. The human body is really complex. We have every cell in the human body that has the exact same DNA, but obviously we have different cell types and cells that are doing different things. So at the RNA level, a lot of those changes are happening. So if every cell has the exact same DNA, then what are the changes that are driving differentiation into different cell types and different functions into our body? So that's fundamentally what we're trying to understand. What are the changes that are happening at the RNA level that are driving differences in gene expression? Well, the vaccine is a very good example of how that might make a difference. So if we can understand how the human body is using these molecules, and we can really do a deep understanding of that. How exactly is it working? What's the mechanism? What's the effect? What diseases does it cause? Then you have therapeutic targets that you can go after. So as a bioengineer, you can harness what's happening naturally and take advantage of it and apply it to something new so that you can develop new classes of therapeutics. Being curiosity-driven is something that's very important. So don't go after it thinking I'm going to invent something. Go after it because there's something that you want to solve. And then just keep going until you solve it. Yeah, I think the students that we have here are really curiosity-driven, and they've been pretty fearless in going after problems that maybe don't seem like they're solvable at the beginning, but once we start working through them and work collaboratively, we're able to do some really cool things here. Absolutely. So CRI has made me, I've always been very interested in technology development, but I'm a bench scientist, an academic. So usually the questions I'm asking are framed around how can we best answer the question. But CRI has helped me think about it in how can we answer that question for us and also develop a product that can be used by the larger community. So it's definitely a different way of thinking. It's the same goal, except it's just a different way of thinking about the problems. We're kind of a startup operation. We've only been around for 4 years, so we're going to continue to develop tools to image and sequence RNA, and we'll see where it takes us. We took a strong turn during COVID. We started working on viruses, which wasn't really something that I had planned. So it sort of depends on the mood of the lab, what talks I attend, people we encounter, and new students that come into the lab with their own questions. So it's possible that in a few years our lab will look very different than what it does today, but it'll always be kind of surrounding technology for understanding RNA better.